Hi everyone! Here's my project on cloning titled Cloning the Hidden Dangers. Hope you enjoy! Simply mentioning the word cloning is likely to generate a debate. The cost of cloning on society cannot be overlooked. It is an extremely inefficient procedure and most clones do not survive. The clones that do survive birth often have mental and/or physical problems. Although cloning animals and humans can have many advantages, the risks and ethical dilemmas are greater than the benefits. In order to allow cloning research to be conducted safely and prudently, more mandatory regulations must be put in place. There are two main types of artificial cloning which could be useful if they were regulated. Artificial cloning includes therapeutic cloning and reproductive cloning. Therapeutic cloning uses a person's own cells to make new bodily systems or organs. According to healthresearchfunding.org, embryos are often used for therapeutic cloning because they have the ability to transform and grow into any type of organ and cell. This type of cloning is potentially useful, but it has not been perfected despite years of research. It also comes with many risks, such as the possible production of tumors. Reproductive cloning is used to create an animal or human with identical genetic features as that of another. According to the UK Biocentre, the most common technique is somatic cell nuclear transfer. This is also known as asexual reproduction. In this process, a nucleus is removed from an egg. Next, a cell from the individual who is being cloned is placed into the prepared egg. Although the clone's genetic features are a carbon copy of the original organism, the clone will not be exactly like the first. There are no guarantees they will look identical due to epigenetics, which is the degree that each gene is expressed. They will also not have the same personalities, as their environment comes into play when determining their temperament. Neither type of cloning has a high success rate. Both types of artificial cloning are controversial due to their unethical side effects. Although there can be many positives to cloning, there are not enough rules to regulate cloning laboratories. Cloning could possibly lead to the development of organ replacement, solutions to infertility, and it furthers genetic research using stem cells. The negative aspects, however, greatly outweigh the positive aspects. Excessive cloning will cause lessened genetic diversity. It is also expensive and comes with ethical concerns. As stated by BiologyWise.com, it is fair to say that cloning can be compared to emulating God. Without increased regulations, cloning will be allowed to go too far. The many cons of cloning outweigh the benefits. There are no laws in the United States that require products from cloned animals to be labeled. The Food and Drug Administration, or the FDA, approved the sale of meat from cloned animals in 2008. As stated by the Centers for Food Safety, the FDA has admitted in its own risk assessment that a vast quantity of animal clones are unhealthy and would not be suitable for food supply. Although both the Food and Drug Administration as well as the United States Department of Agriculture has stated that they would be able to pull these animals from the food supply, they have failed to protect consumers in the past, so their promise is no guarantee. Even if unhealthy clones were not used as part of the human food supply, they would still be used in other ways. These animals, with a few exceptions, are allowed to be rendered into products such as animal feed, cosmetics such as face cream and lipstick, and even pet food, all without being labeled. Like the many that are destined for consumption, cloned animals are not treated well before they die. At the very least, food and products containing cloned animals should be required to carry a label that advises consumers of their contents. Sadly, the current regulations in place regarding cloning are not sufficient. There are no bills that prevent humans from being cloned. The Hatch and Feinstein bills ban scientists from transferring cloned human embryos into a uterus or uterus-like surroundings. Clearly, the regulatory boards consider allowing cloned human embryos to grow into viable fetuses to be unethical. Their loophole allows scientists to create cells with the potential for life which will only be destroyed after their research is completed. In 2003, President Bush stated, "We must not create life to destroy life. Human beings are not research material to be used in a cruel and reckless experiment. These bills do not prevent the establishment of human embryo farms in order to facilitate research. Cloned embryos are created when a nucleus is removed from an egg and replaced with cloned material. While embryo farms may seem unlikely since egg extraction is painful, costly, and time-consuming, it is not hard to imagine embryo farms such as those in the novel Brave New World coming to fruition. Even if embryo farms were only used for therapeutic cloning purposes, millions of embryos, and therefore even more eggs, would be needed. There is not currently an embryo supply this large. In August of 2003, 1,001 adults were asked if scientists should be allowed to use human cloning to create a supply of human embryos that would later be destroyed in medical research. The vast majority, 79.8%, responded negatively. It is clear that most people believe it is wrong to create life and then simply destroy it. In order to abolish questions about ethics, human cloning should be banned altogether. If cloning were to continue without limitations, it would undoubtedly affect genetic variation as well. Many animals have been cloned to increase their dwindling populations. Although this works in the short term, genetic diversity decreases over time. Reduced genetic diversity affects a species' ability to reproduce successfully. This causes the very problem that cloning attempts to alleviate in the first place. Many people are also concerned that cloning can cause mental problems. These mental problems may reduce survival rates, which should be considered in the regulatory process. To date, the high costs of cloning and its low success rate have prevented many of these problems from becoming widespread. The high costs and insufficient results of cloning scream for more regulations. As noted previously, the majority of cloning attempts fail. As stated by the Humane Society of the United States, 99% of cloning attempts fail to produce a healthy clone. This is a spectacular fail rate. Dolly the sheep was the first cloned mammal. It took 277 attempts to clone her successfully. This equates to a success rate of approximately 0.3%. Although cloning has advanced and success rates have risen slightly since Dolly's cloning, the expenses of commercial cloning would only be passed to the consumer, although there will always be wealthy individuals willing to pay the exorbitant costs of cloning. There are little to no regulations on cloning laboratories, and because of the high costs and low success rates, the process must be regulated and limited. As the science of cloning advances, additional safety measures and regulations must be put in place. The use of cloned animals in food supplies and products must be labeled. Human cloning to create embryos with the potential for life is unethical and immoral and should be banned or more highly regulated. In addition, using cloning to breed animals only decreases the gene pool and calls for additional regulations. While there are currently some rules in place, the financial and societal costs are too great for cloning to continue with so little oversight. Ultimately, just because scientists can clone an organism does not mean they should, and they certainly should not have free rein in the field.